All right, let's do this. Today, we're going to take all that genetics theory and turn it into a real practical skill. Think of this as a high-intensity workout, because by the end, you're going to master Punnett squares, one of the most fundamental tools in all of genetics.
You know the answer to that question? It all comes down to genetics, the science of how traits get passed from one generation to the next. So let's dive right in and get a handle on one of the most powerful predictive tools we have.
Okay, so before any good workout, you gotta warm up, right? For us, our equipment is the vocabulary. We need to get a really solid grip on these core terms that, honestly, Gregor Mendel himself gave us.
So first up, alleles. What are they? The easiest way to think about them is just as different options or maybe different flavors for the same gene. So for the gene that controls pea plant height, the options are a tall allele and a dwarf allele. Pretty simple, right? Each parent hands down one of these options to its kid. Now, this next part is absolutely critical. You have to know the difference between genotype and phenotype. The genotype, that's the genetic blueprint, the actual code, like the letters TT or TT. But the phenotype, that's the physical thing you actually see, the tall plant. So the big takeaway here is that the underlying genetic code isn't always what you see on the surface. All right, warm-up is officially over. Let's get into our first exercise. Drill number 1, the monohybrid cross. All this means is we're going to track one single trait, just one, like plant height, and we'll use a really simple but incredibly powerful little diagram to do it. This right here, this is our core technique. Seriously, these 4 steps are all you'll ever need to map out and predict the likely outcome of any simple genetic cross. So enough talk. Let's see how it works in action. Okay, let's put this into practice right now. We're going to cross 2 plants that are both heterozygous for height. That means their genotype is TT, right? So each parent has a 50-50 shot of passing on either a big T or a little t allele. When we fill in the boxes, we get to see all 4 possible outcomes for their offspring. And when you look at the results, it's so clear. If you count the genotypes, the letters, you get a 1:2:1 ratio. 1 TT, 2 TTs, and 1 small TT. But, and this is the important part, because that big T allele is dominant, both the TT and the TT plants end up looking tall. And that's what gives us that classic famous phenotype ratio, 3 tall plants for every 1 dwarf plant. A perfect 3-to-1 split.
Okay, so this brings up a really interesting problem, a bit of a mystery. If you're just looking at a tall pea plant, you know its phenotype is tall, but you have no idea what its genotype is. Is it homozygous dominant, TT, or is it heterozygous, TT? They look exactly the same. So how in the world do we figure out what's hiding in its genes?
Well, Mendel came up with this brilliant solution. It's a procedure called a test cross, and honestly, it works like a genetic truth serum. Here's what you do. You take your mystery tall plant, and you cross it with a plant you know is homozygous recessive, in this case, a dwarf TT plant. And the results of that cross will tell you everything you need to know. Think about it. If all the babies come out tall, then the mystery parent must have been TT. It had no T to give. But if even a single dwarf offspring shows up, you know for sure the parent was heterozygous, TT. Why? Because the only way to get a dwarf plant is if it got a little t from both parents. It's so simple, but absolutely brilliant. All right, you've mastered the monohybrid cross. It's time to level up. Let's move on to drill number 2, the dihybrid cross. This is where it gets really interesting, because now we're going to track 2 traits at the exact same time, say a pea plant seed shape and its color. So the whole key to this is understanding how a parent passes on its alleles for 2 different traits. Let's say a parent's genotype is RRYY. When it makes a gamete, a sperm or an egg, that gamete gets 1 allele for shape and 1 for color. And here's the magic, Mendel's law of independent assortment. Which shape allele it gets has absolutely no influence on which color allele it gets. They're independent, and that means there are 4 possible combinations for the gametes. So if each parent can produce 4 different types of gametes, what happens when we make a Punnett square? Well, our little grid blows up to 16 squares. The principle is exactly the same, you're just combining alleles, but you'll see a really fascinating pattern emerge from the results. I know, it looks complicated, right? But if you just sit down and count the phenotypes, what the seeds actually look like, you will always find this consistent, famous ratio, 9:3 to 3:1. That is the absolute signature of a dihybrid cross. It's the mathematical proof of every possible combination of those traits. Okay, to make sure we're really perfecting our form here, let's go over 2 super common mistakes.
I see people fall into these traps all the time, but the good news is they're really easy to avoid once you know what to look for.
All right, mistake number 1, and this is probably the biggest one, confusing the genotype ratio with the phenotype ratio. You have to remember, genotype is the code, the letters. Phenotype is what you can actually see. And because of those pesky dominant alleles, the ratio of the code often doesn't match the ratio of what you observe.
And the second pitfall? It's forgetting that this whole thing is based on probability. It's just chance. When alleles separate, it's basically a coin flip. A parent with a TT genotype has a 50-50 chance of passing on the big T and a 50-50 chance of passing on the little t to any single offspring. That randomness, that equal probability, is the very foundation that allows the Punnett square to work as a predictive tool.
Okay, you've made it this far. Time for our final challenge. A little pop quiz with a twist. Let's take what we've learned and apply it to a slightly different situation. What happens when you have an allele that isn't completely dominant over the other one?
So this scenario is called incomplete dominance. What that means is the heterozygous version, the RR genotype, doesn't look like one of the parents. Instead, it creates a blended in-between phenotype, in this case, pink. So here's the question. What happens when you cross 2 of these pink flowers with each other?
And the answer is, you get a 1-to-2, 1-to-1 ratio of red to pink to white flowers. Now here's what's so cool about this. Because the heterozygous one looks different, the phenotype ratio, what you see, perfectly matches the genotype ratio. You can literally just look at the flowers and know their exact genetic makeup. No test cross needed.
All right, let's do our cool down. I want you to remember that these drills, they aren't just abstract classroom exercises. Punnett squares, test crosses, these are seriously powerful tools that let us predict outcomes and understand the hidden logic of inheritance. And getting a handle on this logic, that's really the first step to understanding pretty much the entire field of modern genetics.
You see, Mendel gave us this beautiful, simple, elegant framework that explains so much. But what happens when the rules get broken? What happens when genes are linked together on the same chromosome and don't assort independently? Or when one tiny mutation in a single gene leads to a really complex disease? Well, that's where it gets truly fascinating, and that is exactly where we're headed next time.